Thanks a lot, Claire, for the introduction, and of course to the MRC for giving me opportunity to speak to all of you. And today I'm going to talk about two things that many of us find very personal to us. And one of them is the affairs with our gut, and the other is the medications that we take. And the research that I'm going to talk to you about today is going to connect these two things together, and with the hope that we can improve the medications and the treatments in the future. Okay, so microbiome is a topic that's something that does not require much introduction nowadays, and all of us are aware that there are many studies now being published that giving us the importance of microbiome in our body. And I would make an argument that actually we should start thinking our microbiota as one of our organs, and the same way we think about our liver, for example, and we should think about caring for it and protecting it, and also from the medication perspective. And what's the support for these arguments? Each of us is carrying hundreds of grams, sometimes maybe almost a kilogram of microorganisms on our body and inside our body, and most of them in our gastrointestinal tract. And this is a quite considerable amount in terms of the biomass, and also comparable to the mass of some of our own body organs. And it's not only about the number of bacteria, there are trillions of them, but also the number of genes or the diversity of the genes that they host. And this is estimated to be 50 to 100 times more diverse than our own host genetic diversity. And this means that it obviously has a lot of relevance for nutritional, our development and diseases. And there are very great studies that have been published demonstrating this relevance. And one of the things that I'm personally quite intrigued about and interested in is the interaction of the microbiome with the drug. There are studies that have shown that the microbiome is important for a lot of our physiological process. As I said, one largely open question is that what impacts the microbiota composition. So we know it's related, it can impact our mental health, for example, our digestive health, and sometimes also the development and the immune system. So what controls the microbiota composition? Of course, the diet is very important. Other lifestyle factors, the quality of the exercise we do, but something that is actually now emerging now from our another study is also the medications that we take. And this is something that we look quite closely into. And this is what I'm going to talk to you about. So what the drugs, meaning the medication that we are taking to our bacteria. So there are two different ways to look at this, right? So there's one is that cohort studies where you look in or animal models, or you took, for example, large cohort studies where you study thousands of different people between different groups, for example, taking certain one type of diet or the other type of diet, or having type 2 diabetes or not having diabetes, etc. And then you compare these groups in terms of their microbiome composition, and then you can hypothesize that the differences that we observe have something to do with the underlying condition or disease, for example. And there's other way to look at it. So, and why there's need to be other way to look at it. So when we look at this top-down fashion, there's so many confounders. Each of us has a different diet. Each of us has a different lifestyle. We wake up at different time. We drink 1 cup of coffee or 2 cups of coffee. Maybe some of us sick, some never drink coffee. We take medications. We drink diet Coke or normal Coke. So the number of confounders are so enormous that it becomes very hard to connect the observations that we do at the level of the microbiome to the diseases or other conditions or factors that we want to study. So our approach is actually try to help this understanding and insights into this system by studying it from another angle. That's the bottom-up approach, where we want to then take individual bacteria, we culture them under very controlled laboratory conditions, and you can then study in depth what the effects, for example, medications have them. And in the future, then the goal is that to bring this both top-down and bottom-up approaches together, and then they will meet in the middle to have understanding. And one actually analogy in terms of physics would be that if you want to understand, for example, complex planetary motions, we cannot do that without understanding the basic laws of gravity. Right? So we need to understand how two bodies will interact with each other, three bodies will interact with each other before we understand how planets or even complex objects move around. So this is the approach that we wish to take, the bottom-up approach, and then hopefully these two will meet in the middle at one point. So how do we do this? So this is based on largely in vitro systems. Under controlled laboratory conditions, we can culture this bacteria. So we have this resource of around 100 different gut bacterial strains. And importantly, we can grow most of them in a very defined growth media. And this is quite unique, mostly to our lab. Most other places or most other laboratories would actually grow this gut bacteria in a very complex medium with unknown components. And sometimes, for example, some bugs, if you look up into their recipes, you need kind of, you need to add tomato juice and other things that we cannot really scientifically study in depth. And we have managed to culture them with the heroic efforts of the students and postdoc in the lab in the defined conditions, so we can understand their metabolism better. So this is one of the bases that we work with. So what we can do with these bugs. So one of the experiments that we have done, and I will talk largely about, is that they interact with the drugs. So you grow them in anaerobic conditions. So this is where they like to live, or this is where they usually live with very little oxygen. So we grow them in this bags where the oxygen is depleted, and you can then culture them in this microtiter plates in high throughput fashion. You throw different perturbations, for example, drugs or chemicals on them, and you see what happens to their growth, and then you can study this interaction. So one of the first studies we did here, we studied around 50,000 different drug bacteria interactions, looking into how these drugs are affecting the growth of this bacteria. So just to give an idea of how these anaerobic culture conditions look like, here you can, the top left, you can see one of the setups that we have used. So it's basically big, large plastic bag with the gloves inside, and of course an airlock to put things and take them out. And then the conditions inside are made so that oxygen is very little, if any at all. And below there you can see one of postdocs, Sonia Blasher, working in these conditions. And you need to just put your hands through the gloves and you can operate inside, which is a little bit tricky to do, but we have managed to put also some machinery inside where you can automate many of the steps involved. Okay, so what are the results? How do they look like? So this is one of our first results that we published in 2018, and this is effect of around 1,200 different drugs on this bacteria, 40 different strains. That is a subset of the 96 that I talked about. So what kind of drugs they are? There are some antibiotics, but most of them are non-antibiotic, meaning they're human targeted drugs. So they would be, for example, antidepressants, antipsychotics, painkillers, hormones, you name it, really, really large collection of the drugs. And so what do we see? So below you see this big matrix, the heat map over here. So on the left-hand side, this green, mostly green rectangle over here, that's the effect of antibacterial drugs. So you have antibacterial drugs in the columns, and each of the row is one of the 40 culture strains. And if you see a green heat or green color over here, it means that particular antibacterial drug has a negative effect of one of these drugs, right? And this is already quite interesting to look at because these are antibiotics that are designed against pathogens, but they have kind of a collateral damage on our commensal bacteria that are mostly beneficial to us. It is interesting and surprising to some extent to know, but not hugely surprising because they are antibacterials. You would expect that. What was very surprising to us is this middle, the orangish looking rectangle over here. And these are host targeted drugs, painkillers, antipsychotics, etc. And you can see that around 25% of these drugs actually have effect on our commensal gut bacteria. And 25% is a huge number. These are drugs that have been designed to hit targets, protein targets, for example, in the human cells, and they are not been designed to target bacteria. I mean, they negatively clearly affect many of our commensal bacteria, and most of them are against bacteria that are very, very abundant and prevalent in population. So, and this 25% actually is a conservative estimate because we have done these experiments at one fixed concentration. So on the bottom right here, actually you can see the estimated drug concentrations over here. And the one concentration that we have used is the dotted line over here. So many of the drugs actually are exposed at much, much higher concentration. And one of the examples shown here is metformin, which actually has been shown to have a profound impact on our gut bacterial composition. And you can see the estimated concentration that our bacteria in the gut C actually is much, much higher than what we screened at. So it's likely that many more drugs actually would be impacting our bacteria. So at least 25%, I think, is a good estimate. So this, as I mentioned, this is all in vitro study, right? So to say, in a test tube. So how do we know that this is actually relevant in terms of complex clinical setting, or when we take these medications at home, etc.? So we have done that many different ways. One of them is that you can look at the cohort studies where we know what medications people have taken and see whether the effect that we observe in vitro in our laboratory also manifests in those conditions. And all the studies that we have looked at, we do see that effect. Another way to look at it is the side effects. So for many of these drugs, side effects are recorded. And what we observe is that the drugs that we found that have a negative effect on our commensal bacteria, they actually have side effects that they look very, very similar to the side effect of antibiotics. And this is actually another indication and another support that what we actually observe in laboratory actually also manifests in reality. Of course, some of these side effects, for example, diarrhea, one would think that this is not something to worry about. Yes, that would be the case in case of, for example, in life-threatening situations. But if this medication has taken a longer period, some of them actually could become quite troublesome. And there are some drugs, for example, one that's shown here, diacerin, actually it has, and it is used against osteoarthritis. And at least in European medicine agency has actually warned and restricted its usage because severe diarrhea in elderly people and in the negative impacts on the liver. So it can be actually quite life-threatening. So we also, of course, are interested not only in mapping this interaction, but also understanding at the molecular level what's going on. And one of the ways to look at is actually we can look at the gene knockouts. So here, so one of the observations that we have is that there is a kind of a correlation between whether a particular bacteria is resistant to antibiotics and if it's also resistant to non-antibiotic drugs, host-targeted drugs. And they seem to be quite correlated. So it means that if you have a bacterium that's antibiotic resistant, it's likely to be resistant also against host-targeted drugs and vice versa. So what are the mechanisms involved? So one of the experiments we have done is that, so here you see E. coli, that's one of the very common bacterium that or very quite prevalent bacterium that we have in our GI tract. And if you knock out a gene called TOLC, which codes for an efflux pump. So efflux is a process through which bacteria can pump out unwanted molecules, for example, antibiotics outside the cell, and thus protecting themselves. If you knock out this gene, this bacterium, so the knockout strain, the mutant strain becomes resistant to antibiotic as one would be expected. But what we see is that since this moves diagonally up, it also becomes resistant to host-targeted drugs. And this is actually quite an eye-opener because this means that a bacteria can acquire antibacterial resistance, antimicrobial resistance without ever seeing an antimicrobial compound, without ever seeing an antibiotic, only if it sees a human targeted drug as well, because the underlying mechanisms are the same. And this is one of the avenues I think that should be kept in mind when thinking about the drug usage, non-antibiotic drug usage and antibiotic resistance. Okay. And that's one of the lines of study that's being pursued. And if there is another opportunity, I will be happy to talk about that. Now I want to look at the other angle of this interaction. So what I talked about until now is that what the drugs are doing to the bugs. But of course, there's possibility, and there are very good known examples of what the bugs are doing to the drugs as well. And for this, you need to follow not only what's happening to the bugs, how their growth is impacted, but also you need to follow what's happening to the drugs, the chemicals that we're adding to them. And for this, we chose a panel of around 20 different drugs, and there, basically what you see in this plot is showing the diversity of more than 2,000 drugs that are in the market. Right? So what is the chemical diversity? And then we have chosen compounds that actually chemically are representative of different diversities. We also chosen compounds that are very widely used, and for example, antidiabetics, antidepressants, etc., and the ones that also seem to be associated with the gut-related side effects. Okay. So you do the experiments the same as I talked about. You grow them under anaerobic conditions. You add the drugs, and this time you don't only look at what happens to the bugs, but you also see what happens to the drugs. So, and here is the result of that study over here. So what you see on the leftmost side, you see different gut bacteria over here. On the center, you see different drugs, and then the right-hand side, you see the bacteria again. So let me start with the right-hand side of this network. So if you have connection, for example, loperamide, which is an antidiarrheal compound, is something what people take, for example, when you travel to India, etc. And if you have loperamide, if you have this connection over to the bacterium, it means that this drug has a negative impact on this particular bacterium. Okay? And if it's green, it has a positive impact. As you can appreciate, there are very little greens here and mostly red over here. And this is basically what we have seen in the previous study, that many of these drugs have a negative impact on the gut bacteria. So what's new and surprising is on the left-hand side. Now what we are seeing it as a particular bacterium doing something to the drug is depleting the drug from the medium. And there are two different colors you see. You see the blue ones over here. So those are bioaccumulation. I will come back to that very soon. And the other one, purple one, is the biotransformation. So the biotransformation is a process through which the chemical structure of the drug is modified by the bacteria, right? Either to detoxify them or to eat them as nutrients. And this has been actually known for decades. So what was surprising was the bioaccumulation is a process in which the bacteria are soaking up this drug and actually doing nothing to it. And we got so intrigued by this because you don't expect this to happen. We decided to look very closely in one of the compounds, which is Duloxetine, which is marked in the center over here. This is an antidepressant, unfortunately taken by many people, associated it with many different side effects. And as you can appreciate, it's one of the most connected nodes over here. So as I said, it's unfortunately very widely used. So also, understanding what's happening, the effect with the microbiota will be very important. It has GI tract-related side effects, including weight gain. But these side effects and also the effect of the drug is highly variable, and both in terms of dosage requirements and in terms of the side effects across different people. So indicating that there might actually be a significant role played by microbiota in these interactions. Okay, so just to remind, so what is the difference between biotransformation, which you see in the top left, this cartoon over here. So if you have drugs and bugs together, biotransformation is a process where these molecules are modified, for example, broken down or added some other chemical moiety to them. And bioaccumulation is a process where these compounds, chemicals are taken up by bacteria and stayed inside. And it's important to note that in most of the cases, bioaccumulation actually does not affect significantly the growth of this bacteria. And the rest of the slide is basically just showing different assays that we have used. So the bottom left, we see actually the NMR, so that's nuclear magnetic resonance. This is a method where you can actually not only measure the concentration of that compound, but also you can actually ascertain what the chemical structure of that compound is. So we use this method to kind of ensure ourselves that we are seeing bioaccumulation and no chemical modification is happening. And you can do that same with the mass spectrometry, which we have seen, which we see on the right side of this slide. And basically we can recover the drug from the bacterial pellets. You break open the bacteria, you can take the drugs back. So we know that this is bioaccumulation. So what does this actually mean? So the first question we asked, okay, so the drug is going inside the cell, where is it and what's it doing? And what we could do is that we could do some click chemistry to this compound. And the click chemistry is a way where you can add chemical moiety to this compound, and you can then pull down using different methods. And when we do pull down these drugs from the cells, we actually pull down also proteins, many proteins with them, and then we can map them where the proteins come from. And surprising to us, was this many of the proteins that we pull down actually are central metabolic enzymes. And we can also observe that in the neighborhood of those enzymes, there are also many metabolic changes that are happening. The concentration of these metabolites are changing. The one problem with that approach, although quite nice, was that we had to change the structure of the molecule. Right? So we cannot be sure that if the native molecule unaltered, duloxetine would have the same effect. And so we used another method called thermal proteome profiling. So this is a method where you take advantage that if a protein is bound by a ligand, for in our case a drug, its melting curve changes. So when you increase the temperature, the protein gets unfolded, and this unfolding response to heat actually changes if something is bound to it. It can be more stable or unstable. And using mass spectrometry, actually you can do this at the whole cell level, looking at the whole protein. And for this, we chose actually 2 different E. coli strains. So in our setup, we had 2 different types of E. coli, so very related to each other, but isolated from different people. But they had a very different behavior in terms of bioaccumulation. And this is quite important to note because each of us carries a very unique strain of most of the bacterium that we harbor. And this response that we see in terms of bioaccumulation drug interaction seems to be very strain specific. So it will be individual. Every individual would have a different response. And when we do this, compare the melting profile of the proteins between E. coli strains. So on the x-axis below here, you have the bioaccumulating E. coli strain, and the y-axis, you have the non-bioaccumulating one. And if you do this in the whole cell, meaning that you treat the cells with the drug before you do this study, you can see that most of the changes happen in the bioaccumulating strain, right? As one would expect. But if you break open these cells and do the same assay again, these changes, like though both strains start looking very much similar to each other. And what this means is that the difference between bioaccumulating and non-bioaccumulating strain is whether the drugs goes inside or not, right? So and this is very similar to what we have seen before with the efflux pump. So actually the transport across the cell membrane seems to mean quite a lot whether the bioaccumulation happens or not. Okay, another study we did to look into it more biological perspective, it's in C. elegans. So these are worms that are commonly used as model organisms in many of our studies. And they are simpler in terms of study, of course, so makes it easier to study. One advantage they have when studying antidepressant is that you can actually give antidepressant to these worms, and actually you can observe their behavioral change. In this case, so more antidepressant you get, less they wiggle around, right? And this you can see in the lower left chart over here. More duloxetine you give them, so you just need to count how many worms are wiggling around. More duloxetine, more antidepressant, you give them, the calmer they are, and then they wiggle less. And this is a very good then bioassay to look at how much concentration of the drug they're exposed to. Now we can do the same experiment. Another advantage of these worms is that they eat bacteria. So you can give them E. coli that does bioaccumulation and E. coli that does not do bioaccumulation. And here you can see that the bioaccumulating E. coli, which is the blue one over here, actually rescues the effect of the drug, while the other does not do it, right? So it means that the E. coli actually is soaking up the drug, and in effect, the animals are exposed to less amount of drug. Okay. So what intrigued us beyond what the effect, I mean, this is obvious to think about, right? That the drug concentration is changing, but we knew that since these molecules are binding to metabolic enzymes, which are actually quite critical in the life of bacteria for their growth, what else is happening in terms of metabolism? So we suspected that there will be downstream effects. So what Martina, the PhD student who led the study, in the laboratory, one of the very nice experiments she did was that she mixed defined community of bacteria, including some species that did bioaccumulation, some that were sensitive, and others were neutral. And when you grow them, you basically just transfer them from one tube to another tube over 5 days, and you do that with without the drug and with the drug. And you can now appreciate that the composition, final community composition of this bacteria is very, very different without the drug and with the drug. And you can see that the red bacterium over here, that's Eubacterium rectale, is very, very abundant when you have the drug. So this is already interesting to see that the composition of the community changes, but this becomes even more striking when we look at the individual bacteria response to the drug. So if we look at what happens to the bacteria alone. So E. rectal actually is one of the most sensitive and the most sensitive among these 5 bacteria. So in the community context, not only the duloxetine actually changing the community composition, is actually promoting the growth of the bacterium that is actually normally sensitive to it. And this effect is more than simply the reduction in the concentration of the drug, because we see promotion, not only rescue. Okay. So what does this mean? So, it took actually quite a long time to figure it out, but the final experiment that actually quite nicely demonstrated is that you can now grow the bacterium that does the bioaccumulation, in this case S. salivarius, in presence of the drug. You remove this from your tube, you add E. rectale and grow it there. And we follow then thousands of different metabolites using mass spectrometry during the growth of the salivarius, and then later on the growth of the E. rectale. And then you can now appreciate that there are many of these metabolites that are produced by salivarius, and they're taken by E. rectale. And this secretion actually happens much more strongly when you have the duloxetine drug into the medium. So it means that this drug, duloxetine, is creating kind of cross-feeding opportunities. So some bacteria, the bioaccumulating bacteria, are producing compounds that can be used as nutrients by other bacteria, in effect, making the knock-on effect on the community composition. So this is a bit of a complex chart, but what I want to show here is that this is not limited to 1 or 2 bacteria. So we have done this experiment looking into how this drug, in this case duloxetine, changes the secretion profile, metabolic secretion profile of different bacteria. This is in this case 6 bacteria, and you can see in many cases we see that the bioaccumulating strain does the change. And here you can appreciate that this change over here is quite large. And this is quite remarkable, thinking about the phylogenetic and metabolic diversity of this bacteria is enormous, right? So if you think about a microbial community, right, how does one strain pursue other strain? And it's, unless until they're physically next to each other, is mostly through the small molecules like metabolites secreted. So when you add the drug, it would be as if there are different species present in the community. And this effect is also concentration dependent. So more drug you add, more different the secreted metabolite profile looks like. And if you take the same bacteria and then you try to do that with the drug that is not bioaccumulated, you see actually there is not really appreciable difference. The drug doesn't do anything over here. Okay, I think I'm coming towards the end of my part, and just to give a hint, one of the directions we are going towards the future is that, of course, now we know basically what are the basic molecular mechanisms happening. Now we want to build on the complexity and look into what happens in more complex scenario. For example, this is in a synthetic community involving many more bacterial species, in this case 32 different species we are looking into against 30 different drugs and at 3 different concentrations. I won't go into detail, but you see we can actually see quite interesting effects. So these are the drugs over here, different components, and then we look into what happens to the community composition. So we look at the 2 effects. What is the effect of bacteria being together in a community versus being alone, and what is the effect of the drug? And you can see the green effect, all the green colors, dark and light green, those are the expected effects. Right? What would we expect from the individual scenario of the bacterial response into the community? And then this here light blue over here and the red are the unexpected effect, whether the bacteria is protected from that drug in a community or is being sensitized. And we see actually many of these kind of emergent community effects over there. And this is going to be one of my last data slides. So what we can also show that we can again go into these communities and look what's happening to the drug. So this is another drug here we're looking at, not duloxetine, but the story or the idea is the same. We look at what happens to the drug. So we can actually see both drug metabolization over here. So the drug is modified back for some bacteria, but we also see bioaccumulation by other bacteria. So you can see that the both bioaccumulation and biotransformation happening in the community context. So both are very important. Okay, so it brings me to my summary. So what actually do drugs do to our bugs? So there is first, we see a lot of collateral damage to the commensals. So we need to take this into account. Bioaccumulation can alter the bacterial metabolism and really has implications, and of course reduce the drug concentration, has implication for pharmacokinetics, pharmacodynamics, and also in shaping our microbial composition. And we also need to keep in mind that the drug-back relations back to really reciprocal, they both influence each other. And we hope that this fundamental research that we have done, of course, it has a lot of implications. In particular, I think what I'm really excited about is the implications for individualizing the drug treatment in terms of choice of the drug, the dosage of the drug, depending on the microbiota composition. And with that, I would like to end here. And before ending, I would like to really thank many people, and unfortunately could not fit all of them all in this slide, especially in a Zoom context, in a screen context. It becomes very hard to see everyone, but at least the ones that have majorly contributed are shown here. Martina, Sonia, Sergey, Sarela, who have really done most of the experiments into the lab, and also my collaborators and friends from different places, different research institutes. And of course, many thanks to MRC and EMBL to funding this. And with this, I would like to end here.